Thank you. Yeah. This is very much going to be a summary of what we did and a few of the results. I really can't go into too much detail in view of the time, but happy to meet with anybody that wants information or to discuss it further, really. So yes, just to explain, I manage the lab here in Wales. We're one of the partner providers for post-mortem work with APHA, but we do a lot of other things in Wales, CPD, parasitology, and lots of other things. And I guess the reason I show that slide is because for syndromic surveillance, for us to get this to work in Wales, veterinary practice engagement is key. And we're quite lucky in Wales in that we have 2 delivery partners who originally got together to do TB and other services, government services in the UK. And in Wales, we had these 2 groups. Iechyd in the south of Wales, and all of these green dots represent member practices. They actually own the Wales Veterinary Science Centre, so they've got a vested interest already. And so in terms of engagement, we're very blessed with that because they've obviously got a big stake in it. The other delivery partner is Mentor a Business, and this is all the vet practices in North Wales. And whilst they don't have an ownership of our lab, as you see from this next slide, a lot of them are our customers for post-mortem. So these dots, orange dots, represent post-mortems that have come on the carcass transport, and blue dots where the farmers have brought them to us. And you can see that we have a lot of engagement therefore with the vet practices in North Wales and in South. So we're in that privileged position of being able to engage with them. So that's one of the reasons we were asked to help out with this work. So just to put it in context, funding wise, syndromic surveillance in Wales is part of this wider project, RWINE DGC, literally leading in responsible antimicrobial use. That's what it means. And it's all part of this wider project to deliver the Welsh implementation plan for the antimicrobial resistant for the wider plan that Catherine mentioned in the first talk this afternoon. So it aligns itself to the UK plan. And so Welsh Government, through EU funding originally, paid for this project. Lots of partners involved. And you'll see here, if I put the laser pointer on, here we are, part of Iechyd, and these are all the partners that came together for this project. Now, we're just a small part of this project, and there were lots of other things going on. But as I say, syndromic surveillance was part of this. And why is syndromic surveillance so important? Why have we included it in this AMR project? And of course, we want to reduce the necessity or the need or the requirement for antimicrobial use in the first place. We know we can't stop people using it for their animals when they need to, but if we can reduce the need for it, so preventing and controlling disease is key. Also, we can measure baseline of disease. Potentially, we can measure whether things are working, but also to pick up new diseases and give us more data than just the VIDA coding that we currently get with APHA. And we're all familiar with the surveillance pyramid. So for those who are not, maybe, I'm sure most people are here on the conference, but for those who are not, this represents all animals in the national herd. Down the bottom here, we have all animals. Then we've got those with disease, those that the farmers see. Now, the farmer may call the vet out, and then we go up a level, but every time we're getting fewer and fewer animals. The N number is a lot smaller. Then those that are sampled and sent to the other labs. Then we have those that are sent to APHA or SIUC in Scotland, or one of the partner providers like ourselves. And then only then, if there's a diagnosis made, do we get this VIDA data. Now, syndromic surveillance is looking at this lower level, those animals examined via vets in practice to see what sort of conditions they're seeing. And when we started looking at this work, I realized that so many people have tried to do this in the past, people have started, and it's certainly happening with small animal surveillance and a little bit with equine. Both Liverpool University and the RVC in London are doing this quite successfully. But when it comes to farm work, they've not been able to get such data. So why is this? And that was part of the project, really, to see whether we could tap into this farm animal data that's being collected. This example here is just from the SAVSnet, from the Small Animal Surveillance Network, showing how successful this was. And I think this was actually shared in the equivalent conference last year, if I remember rightly, talking about this outbreak of coronavirus in dogs, enteric coronavirus. There was a sudden surge in cases of vomiting in dogs compared to what they normally would see at the time of year, which made them then go looking for a new disease. And of course, that's the benefit of syndromic surveillance. So when we ask the question, what about farm animals? The obvious example would be a case like this lamb here. In 2017, when this came into the lab, I'd already pre-warned and I was suspicious of this having Schmallenberg, having been aborted because of Schmallenberg virus, with deformed legs, deformed shaped head. In fact, the brain was missing when we cut into the head. And this was diagnosed originally in Europe when they saw an increase in diarrhea and deformed lambs, diarrhea in cows and deformed lambs. It made them realize there was an a new disease going on. So they went looking for it, and of course they found Schmallenberg. So that's sort of very much the background. So just explain what we did in trying to see whether we could actually start capturing this data in Wales. So when a vet goes on a farm, vet in practice, he'll examine the animals, he'll make some notes, which he'll record possibly in his day book, maybe a dictaphone into his mobile phone, what work he's done, what he's seen, some clinical notes, and the drugs he dispensed. It then goes on the practice management system so that they can build a practice and they've got a record of what they've done. And so we used two systems to try and get this data, basically the data that's being recorded here. And if I can get that to work, yeah. And we used a system created by Vet in Press, Farm Network, Farm Vet Solutions, Vet in Press. And basically, the farmer records their information directly into a tablet, which captures all the data. The next time they are in internet signal, which in Wales, rural Wales, often it means when they're back in the practice, but when they're back in internet signal, the information gets uploaded into a spreadsheet. Some of it, depending on the practice management system, some of it automatically gets loaded into the practice software, but we were capturing it at this point. The second pilot we used was with Liverpool University FAVSnet, Farm Animal Veterinary Surveillance Network. And this actually captured the data as it was taken from the vet's notebook onto the practice management system. So a slightly different way of collecting it. Either way, the data was anonymized and sent to us for analyzing. Unfortunately, the project took a long time to start, and whilst the whole ROI and BGC project was actually a 3-year project, we were only in a position to collect data for 6 months. So we can't actually draw trends from what we took, but we did collect some data. So I'll share some of that in a second. But from the WVSC, the plan would be then to disseminate that information so that we can have it as early warning systems, so that people can start looking for trends and looking for these abnormal syndromes at different times. So what do we mean by syndromes? Quite simply, we use the APHA submission form, and this is a cattle submission form when submitting samples to APHA, but we used what it's called here, the clinical signs. So we weren't looking for diagnoses, because the minute you look for diagnoses, you're then setting yourself up to fail in some respects, because unless you classify how you make that diagnosis, it's very difficult to say for sure. That's why VIDA coding is so good. So it's very much based on the syndromes. And so we were looking for the primary syndrome. What was the main thing that was wrong with the animal, along with the enterprise type, the age and stage of production, the vet practice potentially, but more importantly, the first part of the postcode, what we call the out code, and then the individual or group problem, so we could put it into some context. And that's the information we were looking to collect. These pie charts just summarize the two systems. So pilot 1 here, pilot 2, the number of animals. The pie charts they're not in proportion to each other. I should have done that. The map on the right shows you where the vet practices were. The darker blue, the more animals from that outcode that we recorded. In the pilot 1, in total, there were 435 cases, but it represented 1,700 animals. And in pilot 2, 392 cases. This was November to May, so November 22 to May 23, and this was February to April 23. So peak lambing time, and that's important. You'll see that in a second. So this is quickly how the first pilot worked. The vet has this screen on their tablet. They can choose their animal. In this case, we don't know, so we click unknown ID. We select how many animals, what stage it was. So in this case, we're going to select a breeding bull. And then over to our right, we click once for the primary syndrome, and then we click twice for any other syndromes that we've got. And this is just a summary over the whole study. There's no time to this. We can't draw conclusions from this, but you get an idea of the primary and secondary syndromes that we saw in cattle, the blue bars, and the primary and secondary syndromes we saw with the orange bar. So that's the first system. Very conscious of time, so apologies for flying through. This is the second system where you actually put it straight onto the practice system. The vet enters the farm details. This screen comes up. They first of all have to click which of their syndromes it is. They then get some follow-up questions. So species type, enterprise, what stage of production, and then they work record as they normally would, visit, drugs dispense, what they saw, and we get that data. And rather neat with the FAVSnet system is they use the Tableau software, which is, I believe, the same software that APHA uses for its dashboards. And we can display the information this way, even to show the number of consults for a particular condition that required antibiotics. So we could click on diarrhea and see how many of them required antibiotics. So again, quite useful when we're looking at which diseases or syndromes to look at when we're trying to reduce antimicrobial resistance. Sorry, mouse has gone crazy. So that's antimicrobials. And again, this is a summary of the syndromes according to species that we saw with pilot 2. Just want to share another graph, and apologies for all these graphs, but I think they're quite good at showing this picture. The other thing we did is compare the syndromic data. So this is all the list here. This is pilot 1 and pilot 2 with the animals that we were seeing at the WVSC here that were coming in for post-mortem and what syndromes they were showing prior to their post-mortem. And you'll see practice pilot 1 and 2, very similar color distribution, really completely different to the lab. And it's quite useful for this because it does show us that if we were to do syndromic surveillance based on animals submitted to a path lab only, then we would certainly not necessarily have a fair representation of what's seeing in practice. But I mentioned the time of year when this was done, and this data is just from February to April on both pilots and in the lab. And you can see in pilot 1 and 2, reproduction was one of the main reasons that vets were seeing animals, and that makes sense. It's lambing, it's calving season. So what is the corresponding one in the lab? Of course, it's abortions. We were getting fetuses in to find out abortions. So although they're different, you can see there are some correlations there between them. And that's quite rewarding that we were seeing what we were hoped we would see at this time of year. Submission rate throughout the pilot. This is all practices. So 3 practices submitting to pilot 1, 3 to pilot 2. There's a sudden surge in pilot 2 data here. This was a third practice that joined us late into the pilot and changed their system completely to record the data for us. But we know it wouldn't have been a long-standing solution. So it just shows you that we do get ups and downs when we get the data in. Our biggest problem was engaging vets to actually do this work. And so just some conclusions I'm going to draw very quickly. It is possible to collect this data in Wales. However, it requires a cultural shift in the way vets work, and vets are creatures of habit. The data we can collect from the practice does differ from the diagnostic labs. Vets won't collect the data if it's too complicated or it takes too long. They're too busy. There are too many management practice systems for us to have a one-size-fits-all approach for all practices. And the question is, do we actually need to do all practices, or could we just use some geographically spread around Wales to be representative of what's going on? What's next? As of, I think it was yesterday, we got ethical approval to use FavsNet again for a bit longer, what we call FavsNet Lite. This is going to basically be a passive thing in the background. So vets will record as they normally would without any of those additional screens. And it will pick up the data from what they're writing. So it will be looking for keywords. It will be text mining. We're going to use the existing practices. So I say the ethical approval is now in. We're going to try and recruit more practices, and then we're going to explore the quality of the data as we go forward. Apologies, I went slightly over time, but just like to acknowledge the vet practices and the different organisations that helped us with this project. If people are interested and want more information, then please do get in touch. Thank you very much.